Hello everyone, welcome to Chemical Chemistry 256 experiment, organic chemistry experiment. Here in this video, we'll be looking at identification of an unknown aldehyde or a ketone. First, let's look at the objectives. In day one, this one will be performing in 2-day experiment. Day one will be looking at simulation. Each unknown is assigned to you and renew the screen to see the unknown number matching the number the unknown assigned to your group. So we'll be doing the simulation and identify unknown aldehyde ketone by using chemical tests. We'll be using two chemical tests. First one is Tollens Test. Second one is Iodoform Test. And second, looking at IR spectroscopy, and especially looking at the ketone. Our unknown is either aldehyde or a ketone. So we'll be looking at the carbonyl peak around 1700, and we'll be using the peak is greater than 1700 per centimeters wave number or less than 1700, and we'll be able to tell the resonances of the carbonyl compound. And finally, we'll be looking at the boiling point. It's not easy to really determine the boiling point from the simulation, but I'll be carefully looking at the boiling point and record the boiling point. And in this way, we'll be able to identify the unknown. Our unknown is one of 6 carbonyl compound given in our lab manual. And then also make sure to fill up the tables 1 and 2 because we need the properties, and these literature properties can be used as a reference for comparing our data with the literature data. So when we come back on day 2, we'll be using our unknown and synthesize 2,4-dinitrophenyl hydrazone, and this one has a solid. And then so we'll be using our basic organic chemistry techniques, and we'll be recrystallizing, we'll be dry it and measure the melting points and compare with the literature value. It'll be really cementing our identification of the unknown. Okay, so now let's look at the details of the compound. So the structure of unknown. See, unknown is one of the 6 carbonyl compounds. So it is 2 aldehydes and 4 ketone compounds, and so it is the benzaldehyde, cyclohexanone, acetophenone, 2-heptanone, 2-octanone, and heptaldehyde. So either this kind of 6 or 7 or 8 carbonyl compound, and so they have different properties. First, let's look up this aldehyde. So we have 2 aldehydes right there, and the remaining 6 of them, they are ketones. Aldehydes will be positive for Tollens test. So these aldehydes will be positive for Tollens test. So it will be generating silver. It will be producing silver. Silver will be produced, and the color of silver is a dark gray color, dark gray precipitate will be formed. And so look up the chemical reaction for the Tollens test part, and if the test is producing dark gray precipitate, then this is positive for Tollens test, and the compound is either benzaldehyde or heptaldehyde. If not, then it is negative, then it is either cyclohexanone, acetophenone, 2-heptanone, or 2-octanone. And now let's look at the second criteria. Second criteria is iodoform test. Iodoform test is positive for methyl carbonyl, methyl ketone, especially in alpha position. For example, when you have a carbonyl compound, next carbon is called alpha, the following compound is beta and gamma carbon in this order. And cyclohexanone in alpha position does not have methyl, so this one will be negative for the iodoform reaction. Acetophenone, by the way, has methyl group right there in alpha position. This will be positive for iodoform test. Iodoform test. Let me erase this one. Let me erase iodoform. Okay. So it'll be for iodoform test. It'll be generating yellow precipitate. So when you see in iodoform test, the reaction generates yellow, then it is either acetophenone. We also here has heptanone and octanone. They all have in alpha position methyl group, alpha methyl. So when it is positive for iodoform test, we are narrowing down our candidate is either of these three compounds, acetophenone, 2-heptanone, or 2-octanone. And then we'll be also looking at the IR spectroscopy. And now let's look at benzaldehyde and acetophenone. Benzaldehyde, acetophenone in the carbonyl compound is benzylic position. So for example, here in this one we'll have resonance structure. This one will have resonance structure, and it'll be forming positive charge on the benzene ring, and we'll have this alkoxide will be forming. This alkoxide is forming, it is part of the resonance structure. Then carbonyl compound, it has less of double bond characteristic because of this resonance structure. So then in the IR, the peak will be CO double bond stretch will be found weaker, meaning it will be found in less of 1700 wave number. So then the IR peak, the carbonyl peak is either 1680, 90 region, less than 1700, and it is either acetophenone or benzaldehyde. So we can use those IR peak to identify it. Finally, looking at the boiling point, and we can really finalize our identification. So boiling point, compare with the literature value, and then determine our identity. Okay, so then next one is each of the test, Tollens test, identify an unknown. So we'll be preparing Tollens reagent. First, silver nitrate solution will be treated with a strong basic condition, sodium hydroxide. It will be forming silver oxide, silver 1 oxide, silver oxide, and this will be solid. And then we'll be adding ammonia solution, and dropwise add, it will be forming a silver, so dinitro-argentinum hydroxide, diimine argentinum hydroxide. This complex will be forming. So silver oxide will dissolve into solution, will be forming this complex. And then once this complex formed, let's add our aldehyde. Then here in silver is positive 1 charge, and it will be reduced to silver metal, and it will be forming acetate ion. This acetate ion, acetate ammonium, and it will be generating 3 moles of ammonia. So this one, silver mirror, this color will be dark gray color. So then you see dark gray precipitate is formed. That's a positive for this Tollens test, and it is either, our unknown is either benzaldehyde or heptaldehyde. Next, ketones do not react in Tollens test. Only aldehyde. Aldehyde is more reactive than ketone, remember. So then only aldehyde will be positive in Tollens test. When it is negative for Tollens test, we can see that it is one of the 4 ketones. Now let's think about iodoform test. This is identify for methyl ketones. Step 1, halogenation of methyl ketones. And we'll be soon learning about alpha carbon chemistry. So here is carbonyl compound, and next position alpha. The protons in alpha carbon is very mildly acidic, pKa 20. That's the difference between other carbons in alpha carbon. This is very important for alpha carbon chemistry, enolate chemistry. So in a strongly basic condition, and it'll be taking, it'll be in equilibrium, but it'll be taking some of this acid, forming a deprotonated, forming an enolate. This enolate will be reacting with iodine, and then it'll be forming iodination occurs right there. When you have a monoiodination, then this one is more acidic because this is electron withdrawing group. Iodine is stronger in its electronegativity than carbon, so this is forming an electron withdrawing group, and then this hydrogen becoming more acidic than alpha carbon. So the reaction condition is highly basic condition. Then hydroxide will be more readily taking hydrogen, deprotonating, and second equivalent of iodine will be added to the carbon, and it will be going one more round. Finally, all of the hydrogens in the alpha carbon will be substituted by iodine, and this one is taking iodine is taking electron density away, and also iodine is very bulky. So then when all of the three hydrogens is substituted by this iodine, this triiodocarbon becoming good leaving group, and this is next to the carbonyl compound. So then nucleophilic substitution will occur. Okay, alpha hydrogen of carbonyl is acidic. This is a key student learning objective in the next part. After carboxylic acid and the derivatives, we'll be studying about alpha carbon chemistry. So this is a core student learning objective. pK value of 1920, derived alpha carbon of a ketone. So then when we have a good leaving group, then hydroxide ion will not functioning as a base. This will be functioning as a nucleophile. Hydroxide ion is strong nucleophile, so it will be attacking carbonyl carbon. It will be forming tetrahedral intermediate, and tetrahedral intermediate, the carbonyl can be restored, and this one good leaving group triiodocarbon methyl will be leaving. And then next, this one carbon has now carbanion characteristic. Remember, carbanion is very reactive and unstable. Usually they do not leave, but because of this iodine, 3 of iodine group is taking electron density away. At the same time, they are very bulky. So these two factors combined together, making this triiodomethyl is a good leaving group. But still, because of a lone pair of carbon-ion characteristic, this is strong base. So it will be taking the carboxylic acid is very mildly acidic. I mean, good acid. pK value of around 4. So it will be taking proton away, forming iodoform and acetate ion. Here, acetate ion, and it will be forming iodoform. And iodoform is insoluble. The carbon ion is soluble because of negative charge in acidic condition, but iodoform is no charge. So this is precipitating out of solution, yellow solid. So then yellow precipitate forms, that's the positive test for iodoform test. But the thing is, this is a little bit tricky to identify the yellow solid. So then please take attention to see yellow solid is formed in our simulation carefully. Okay, sometimes we are missing the formation of yellow solid. The yellowness is faintly yellow. It's not vividly yellow. It seems to be close to the white. Okay, so then be careful to look at this yellow solid, and then it'll be showing that the formation, the location, the presence of methyl group right next to the carbonyl, alpha methyl carbonyl. Okay, so next, determination of boiling point. This one is set up for determination of boiling point. We'll have a test tube in like our liquid will be inside there. Our unknown liquid unknown in the test tube. And then test tube, right next to it, we have a thermometer. Here we have a thermometer outside. They are bundled together with a rubber band. And then a test tube, capillary tube is now inverted inside into our liquid unknown. Okay, then let's heat it up. Let's raise the temperature. Starting from room temperature, heat it up slowly. 30 degrees, 40 degrees, 50 degrees, 60 degrees. At certain point, you will see bubbles coming out. Bubbles coming out from the capillary tube. Okay, and then the formation will be slowing down, and liquid from our liquid unknown, liquid just enters the capillary tube. That's our boiling point. And then read the temperature. Read the thermometer. That's our boiling point. Okay. One more time. So then this equipment is set up. Our liquid unknown is in that test tube about like half level, less than half level, and the capillary tube is inverted down, and thermometer is bundled together by rubber band. We are heating up. At a certain point, nothing will be changing, but at a certain point, bubbles will be coming out from our capillary tube, and after the bubbling all out, liquid will be started to enter that capillary tube. That's the boiling point. Okay, so then read the temperature of the thermometer. That's the boiling point. And then compare with the literature value. This will be really confirming the identification of an unknown. By using these chemical methods, and we were able to, see, narrowing down our carbon is either aldehyde or ketone. The ketone has methyl group right next in alpha position, and we have multiple candidates. And then reading this boiling point, then we can really narrow down our unknown to something, a one compound. Okay, so liquid just enters capillary tube. That's the boiling point. And now in day 2, so also IR data is given right there. IR data given in the simulation. We'll be reading also IR data, and then you can tell the formation of benzylic position. The ketone, the carbonyl group is in benzylic position, then it'll be forming a resonance structure. Then the carbonyl IR peak will be found under 1700 of the wave number. And so when it comes back on day 2, we'll be doing this formation of 2,4-dinitrophenyl hydrazone reaction. General reaction scheme is aldehyde or ketone, and we have hydrazine right there. So we'll be forming hydrazone reaction right there. And then when you have hydrazone, like acidic condition, and then we can separate them out. This one, we have seen this mechanism many times in our lecture, and it should be an acidic condition. We learned that the best condition is pH 4 to 6, and we are using relatively good acid, but not that strong acid. This phosphoric acid pK value is pK1 because we have 3 of the proton that can be deprotonated. pK1 value is 2.2. Okay, so compared to other strong acids such as hydrochloric sulfuric acid, their pK value is not negative value, -2, -7, very strong. And this phosphoric acid is a good acid, providing acidic condition, but also it's not at the same time not a strong acid. So it'll be providing the acidic environment that is necessary for this like hydrazone formation reaction, iminium ion or imine reaction. Okay, so let's look at, so this one will be solid. Will be separating this one solid, and then we'll be using recrystallization to purify of this hydrazone compound and determine melting point, comparing with our literature value, and this will be a final confirmation of the identification of our unknown. Okay, melting point of hydrogen derivative is different, like our 6A carbonyl compound, their melting point of hydrogen derivative is all different, so then we can really picking up, finally find out our unknown. And so the mechanism is very similar as we studied about iminium ion imine formation. So hydrazine, we have this nitrogen single bond, we call it hydrazine, and this hydrazine derivative will be in, this one is mild base. So in phosphoric acid, the majority of them will be forming this protonated hydrazine. But still, because phosphoric acid, as I said, the pK1 value is 2.2, and this pK value is around 9 to 10. So majority of them will be forming the weaker acid, the product side, but still good enough amount of the compound will be remaining, hydrazine. Remember then in acidic condition, we have acid, this acid, and this hydrazine compound, both of them present. Okay, majority of them will be acidified, protonated hydrazine, but still good amount of the compound is hydrazine, the reaction will go through. So now let's look at it, the reaction mechanism. So please draw this reaction mechanism in your lab notebook. First one is this nitrogen is like good nucleophile, not strong, but good nucleophile. So then nitrogen will be taking carbonyl compound forming tetrahedral intermediate. Now this is acidic environment. Then the tetrahedral intermediate will be protonated. So protonation right there, protonation, and then deprotonating will be deprotonating. Finally, we need to convert this poor leaving group. So hydroxyl group is poor leaving group. Let's protonate it because it is acidic condition. We'll be using like large amount of phosphoric acid, excess amount of phosphoric acid. It will be protonated, and it is now good leaving group. It is oxonium ion is good leaving group. Then the lone pair of nitrogen will be coming down. I describe this one as a pushing button. The good leaving group is ready to leave, but this tetrahedral center is highly statically hindered. So then, outside nucleophile cannot attack. So then the lone pair nitrogen must be coming down, forming iminium ion, and then water will be leaving. So loss of water is a major mechanism of this process, and it will be forming iminium ion. This hydrogonium ion will be formed, and it is reactive chemical species, and then it will be deprotonated. So then deprotonation here, proton transfer, deprotonation will be occurring, and finally we'll be forming hydrazone. And then following this one will be recrystallizing and determining the melting point. So follow this one, we'll be doing recrystallization using alcohol. Okay, dissolving this one in alcohol, and then determining melting point. When we come back on day 2, we'll be doing this chemical reaction. Okay, so this reaction is very effective reactions. We'll add excess amount of phosphoric acid, and we'll be adding 60 microliters of our unknown liquid, and then just heat it up, and then the reaction will be achieved within 10 minutes typically. And then we'll be doing recrystallization and then making them into a solid product, and let them dry, and then determine melting point. That will be day 2. Okay. So then CO stretchy, CO compound conjugation with other pi bond. The CO bond is weaker. For example, acetone. Acetone has a methyl group right there in the alpha position, so it is isolated, no resonance structure. But in benzophenone case, in alpha position, we have, this is a benzylic position, benzylic ketone, so we can draw resonance structure. So then the pi bond, the CO bond is weaker. CO bond has more single bond characteristic, single CO bond. Because the carbonyl has now more single bond characteristic, it's it is weaker. It is showing in IR peak less than 1700. Remember, this is less than 1700 of wave number. So that's the evidence that we have this benzylic position. So then look at the CO double bond stretch in IR spectrum. If the bond is less than 1700, the peak is found in less than 1700, then it is one of the two compounds that is in the benzylic position, carbonyl in benzylic position. Okay. Now let's look at how to determine chemical reactions. And I mean day 2, this one will be day 2. When it come back in day 2, we will be following this process. Set up a hot water bath. We need to heat it up, this one, the reaction, because nitrogen, the carbonyl of nitrogen in the carbonyl is not very strong nucleophile. We need to heat it up, making the reaction going. And hot water bath will be adding 5 ml of 2,4-dinitrophenyl hydrazine stock solution to a centrifuge tube. Okay, using centrifuge tube. And here, the solution contains phosphoric acid. The stock solution already contains phosphoric acid, and that is extremely corrosive. So make sure wearing personal protection gear, wearing gloves, wearing our lab goggle, make sure. And then adding 60 ml. 60 ml of a assigned unknown. Okay, assigned unknown using pipette, the auto pipette. So this one will be the first time we'll be using auto pipette. In biology, we'll be using microbiology, we'll be using auto pipette almost every day. But in chemistry, this is the first time to measure out 60 ml. And so I'll be demonstrating how to use the auto pipette when you do a day 2 experiment. And assign, measure out, and then adding it to the centrifuge tube. Now it is mixed. Next is heating up. The chemical reaction hot water bath. Clamp the centrifuge tube in hot water bath. Okay, clamp it to fix the position, its stationary position, and heat it for 10 minutes. You see the crystallization of hydrazone product. So hydrazone product is highly we see nitrogen, and then long like carbon chain. It is very insoluble in aqueous solution, so it will be precipitating out of the solution. You see solid product will be forming. And troubleshooting, if we do not see crystallization after 10 minutes, then heat it up a couple more minutes, then usually gives crystals. Let's do recrystallization. Remove the tube from the hot water bath and allow it to cool to room temperature, and then let's separate it in vacuum filtration. So vacuum filtration, let's filter it out. Wash with cold water. So rinsing with cold water. Remember, we have used excess amount of phosphoric acid. It is important to rinse it out, the acidic condition making it to neutral condition. So then wash with cold water 3 times with 5 ml of cold water. And then let's remove all of the remaining phosphoric acid. It is important to rinse several times with cold water to make sure to remove of acid as much as possible. Okay, and so let's use pH paper, litmus paper onto the crystals. Touch it. Okay, press it. See if the crystals is showing it is acidic condition. Okay, if it is acidic condition, add more additional 5 ml of cold water. Rinse it a couple more times until no longer is acidic. Okay, this shows that really the making, washing, rinsing is very important. Okay, and then scrape it to a 10 ml Aldehyde flask. Scrape the product, and then let's do recrystallization. So then put it into a hot water bath. Okay, set up in hot water bath. Add this 10 ml Aldehyde flask and put it into hot water bath, and also making ethanol hot. Prepare ethanol hot. Remember, the boiling point ethanol is 78 degrees C. So then do not boil the water bath. Okay, so near 90 degrees C, that's good enough. And this hot water bath added little bit to the aldehyde flask, the crude product in the aldehyde flask. Use minimal amount, little bit, and then there is the aldehyde flask is in the hot water bath. Okay, and add a little bit until it is fully dissolved. And use, make sure to add a minimum amount of hot ethanol, because we want to have maximum amount of yield. Okay, do not add too much, little bit until it is completely dissolved. And then take this one out, aldehyde flask, put to the air, and the room temperature air, and then let it cool slowly. And once it reaches room temperature, and further cool in the ice bath. Prepare ice bath. Put it into the ice bath, the aldehyde flask. Let it cool the ice bath, and then do vacuum filtration one more time. And wash with cold ethanol. Okay, cold ethanol. And finally, we are ready now. Let it dry. Scrape the product onto watch glass, dry in the air, like let's say 10 minutes, something like that, and it'll be completely dry. Ethanol will be evaporating into the air. And we are ready now to determine the melting range of solid hydrogen derivatives. So then use a melting point, the capillary tube, and put it into the melting point, the machine, and then let's read. So the identity we already know identity by TOI interactive activity, and compare our melting point that it is written in pre-lab table 1. So compare the value. And we know that this literature value, so then our melting point should be 30 degrees below, starting 30 degrees of below, starting point, start, and 20 degrees high. 20 degrees is higher than the literature value. And then make an observation in this window, temperature window, see if the melting point, so starting point or melting point, remember melting point already in a range, it's not a single point. Okay, so then determine the starting melting point and end the melting point, record it in your lab notebook. Okay, thank you everyone for joining me in this very important activity of identification of unknown aldehyde or ketone. So in day 1, by using this online activity, by looking at this simulation, then we'll be able to identify unknown. In day 2, we'll be coming to the lab and then converting into a hydrogen derivative, and we'll be perform recrystallization and determine the melting point, and finally compare our literature value. Okay, thank you everyone for watching this video. I will see you in the lab.